It ends today at 4 o'clock, the judging. I'm not sure if you've had a chance to go look at the art, but where the registration area is, there's a group of windows. There's easels there. Please look at the art and put in your ballot. And the announcements will be made tonight at the Rock and Roll Hall of Fame, so I hope everybody's planning to come to that reception. It begins at 6:30. In your badge, there's a gold wristband. Make sure you have that on. It's just a little paper band, but they won't let you in without that, so fair warning. Wi-Fi, just a reminder for anybody who's just kind of showing up today, the access code is ASGSR2016. We put a lot of thought into that one. And then last this morning, 2 student posters after this session. Student posters are going to be in the next room, so please chat up with our student scientists, our young scientists. So with that, I would like to get the session started. I'm introducing Dr. John Olson, if he'd like to come to stage and begin the panel. Thank you.
Okay, good morning. It's nice to see so many shiny faces out there. We have we have a World Series event for you right here in this town with this panel. I think we got a great lineup, and I think hopefully you'll find it riveting. We are going to bring up the slides here. All right, is somebody turning these on for me? Okay, sorry. There we go. My bad. Awesome. Well, I'm John Olson, and I would like to shed a little light on this great panel that we have for you. It's Research Enabling LEO Commercialization. And as we're putting together the panel very briefly, just take a look at all those sponsors up there, and I'd like to thank each and every one of you who is here representing one of those sponsors. But what's super neat about that in the context of this panel is if you look at the richness and diversity of just those names and those companies, we have quite a broad spectrum. We've got some old familiar names, and we've got some new, fresh ones, and that is a very healthy mix, and I think is indicative of the richness with which our microgravity research efforts are taking place within the commercial context. As we look at our panel today, I'll be your moderator. Essentially, I'll set the context for the broader panel, and then when we have 4 all-star members in our lineup here. We got Dr. Joe Eng, who's from iExpress Genes, and we also have Dr. Supriya Jaswal from Astrolux, and we have Dr. Alexey Churilov from Radiation Monitoring Devices Incorporated, and finally Dr. Roz Smith from Eli Lilly and Company. And so we'll bring those. We'll bring our panelists up to the table. I'd like to give you just a brief overview of each one. Dr. Joe Eng is the director of the Biotechnology Science and Engineering Program at the University of Alabama, Huntsville, and he is the principal investigator for research team studying structural biology there. He's also the co-founder and president of iExpress Genes. Now, Supriya, she is the CEO and founder of Astrolux Corporation, Astrolux, okay, if we pronounce it the French way, but it's Astrolux Corporation, which is a next-generation semiconductor company in San Diego, and they design advanced engineering technologies that enable high-volume manufacturing of next-gen IC, integrated circuits, at the 14-nanometer and smaller range. Dr. Alexey Churilov is the senior scientist at Radiation Monitoring Devices Incorporated, and they specialize in developing new crystals for radiation detection. And finally, Dr. Roz Smith, she is currently a research fellow at the Biotechnology Discovery Research Component of Lilly's research laboratories, and she works in she leads the regenerative biology group, which identifies bioproduct candidates for clinical development. So I think what you can see is that's quite a nice portfolio of speakers that we have for you today. So let's put it in context. Let's look at the space value proposition for microgravity research. Right now, for economic value to the country, aerospace represents about $2.5 trillion if you look at the direct and derivative benefit. Roughly aviation is about 1.43 trillion of that, another trillion in space. Indeed, it's largely the reason why we have a positive trade balance in that sector. 500,000 direct jobs, 750,000 indirect. So this is a huge engine of economic activity and innovation. Similarly, if we look at the commercial space context, and we look at this is a Space Foundation report from 2015. If we look at overall the global space activity and the revenue breakdown, over 75% of a $330 billion annual investment, or annual expenditure, I should say, is commercial space related. Similarly, about 2/3 of the launches are for commercial purposes. And when we look at the global economic picture, it's been roughly flat at maybe 2% of growth. But in this sector, again, we're seeing sustained growth at 7%, almost tripling the global sector. And finally, satellite commercial activity is very up. Right now, and I say right now, and I qualify it as currently, the United States is the global leader in aerospace. And to continue to advance and sustain this is going to take diligent, focused work. A little bit more factoid information for you. Since space, 1.8 billion, since 2014, companies have raised more than 1.8 billion, and these are space companies, so much more in direct and derivative. And as we look at just the explosive growth in nanosats and small sats depicted there, we expect by 2020, 2,000 plus projected to launch. $5 billion in commercial satellite imagery revenue, 56% through 2016 of nanosats will be used for commercial use. So that's more background about the commercial viability of low Earth orbit and beyond. But what is LEO commercialization, low Earth orbit commercialization? As we look at the economic impacts, those are clearly set as we've just walked through. It's also a huge driver for security. It's an enabler for productivity and really freedom of opportunity, freedom of action, and freedom of growth. In largely the technology and the innovation areas, and those feed exploration, both terrestrially and beyond LEO. But it's also about sustainability, the benefits for our planet, the technologies, the spin-offs, and the spin-ins. So breaking it down and looking at the numbers, let's see this broader context. The United States government peak funding for research and development in the history of our government was in 1967, wherein it comprised 67%, 2/3 of what was spent. But interestingly enough, if you look at the current picture today, 2/3 of that, at 63%, is private R&D. So we've had a flip-flop, and so largely that was that public sector funding for R&D was post World War II, and it is wound down. Currently, U.S. government basic R&D funding represents the largest segment of our $66.1 billion annual investment, 36 billion of which is U.S. government basic R&D, wherein private industry only funds 20 billion of the basic. When we look at the global context, the United States is leading global spending at 429 billion, but if you look at the European Union, the integrated composite picture, 320. China, in just the past 10-year period, or the 10-year period from 1991 to 2011, 27-fold increase. Japan doubled it during that same period, and South Korea has increased it 8 times to 60 billion. So these are critical for leadership in space and elsewhere, but I think this shows the globally increased focus on these areas. And when we look at within the United States in terms of the low Earth orbit economy stimulation, we spent $14.2 billion in NASA since 2005 on the commercial orbital transportation services, the CC Dev, the Commercial Crew Transportation Capabilities Program, and then Cargo Resupply 1 and 2. We spend about $3 billion a year on ISS operations and several commercial initiatives, some of which Nanoracks and Bigelow would be exemplars of. But what has been the result of this is we've dramatically increased our access to space, or our space transportation capabilities, and you see those companies and their products listed. But we've also established cases to really steward the 50% of the U.S. operating segment, and yet, despite all those expenditures, we still have some challenges for a truly sustainable or self-sustaining low Earth orbit commercial space economy. So this gives you just one snapshot, and I pulled from SNC, which is something that I'm very familiar with. But for ISS, the up mass and down mass is fundamentally important for R&D advances, and really a diverse ecosystem, one which has similar redundancies and resiliency, but these abilities below, the capability, the affordability, reliability, reusability, and operational utility, those are vital to make sure that in the R&D and the commercial sectors, we have these as trusted transport to and from. Well, the benefits are clear. Geopolitically, it allows us to focus on the beyond LEO and focus on the inherently government core activities. It allows us to have regular and assured and operationally flexible access to space. It allows us as an export benefits, and the intangible part of generating new scientists and engineers and creating this pipeline, STEM or STEAM pipeline, is so important. Compelling missions, compelling work, and compelling economic opportunities drive that. And finally, it's the diversity and quality of the outcomes in the research and development technology solution areas that really lead and are the ecosystem. But that ecosystem is a role filled by government, interested investors, and companies. And today what we have for you is, as we look at the International Space Station, the whole context for this panel, and we look at the commercial companies, really vibrant examples. We've established that transportation is key to doing their great work. But these R&D pathways that each is setting up in part of the broader ecosystem are really the key to the next giant leap. So for today's panel, we're going to talk about how we're using this ISS National Laboratory, and we're going to talk about the exciting research done by these truly commercial companies, and we will talk about how that is a critical part of the overall ecosystem. And that ecosystem will feed our beyond LEO exploration, our new capabilities, and our innovation, and it comes from all the sectors. And in this year of big politics, I think the key is that we need cooperation. That's bicameral, bipartisan, interagency, international, industry, and academic. That's the key. And we need to have this as a national priority. It's vital to success, and these sustainable commercial activities are the core and vital elements of it. So we're at a new era. This is the dawn. Just like today is the dawn of a great day, so too do we have lots of opportunities in civil, commercial, and national security space. We have to be bold and visionary. Capabilities, as we've invested and stimulated and developed, are enabling, but they're necessary but not sufficient. We are going to be highlighting today need to be focused in a competitive commercial sense. They need to have true sustainable value. I think harmony, and go through some of these divisive discussions, harmony is the key in a balanced and affordable and an efficient ecosystem that works. And if we don't make ours work, the global environment will yield winners and losers. And finally, I think vigorous pursuit of these research pathways is the way to go. So with that, I'd like to bring up our panelists, and we'll go through today's panel. First up is Joe Ng.
Good morning. Thank you for making it this early in the morning. I'm very excited here to tell you what we've done, the experiments we've performed using the ISS growing protein crystals for applications in structure-based design. So to set the stage, I want to explain to you what is structure-based drug design. So when you have a drug out in the market, there's a lot of work that's done prior to that. And one of the most important things is specificity, meaning that is a drug specifically effective for a certain disease. So the pathway for structure-based drug design is the following, is that you identify a disease that you want to attack, and in this case, maybe a foot disease or some kind of infection, and then you look now at genome sequences or protein sequences that you may be able to produce the protein and try to block its action that may kill the bacteria that's responsible for its ailment. Well, in our case, one of the criteria, or at least the bottleneck step, is to crystallize these proteins. And the reason for this is because crystallizing proteins is the barrier for a technique called crystallography, which determines the outcome of the three-dimensional structures. Three-dimensional structures is important to identify definitive locations of atoms in a protein. And why is this? Because if we know the exact positions of proteins, if we know the exact architecture of a protein, then from an engineering perspective, from a pharmaceutical perspective, we could design something to block it specifically. And by virtue of blocking it specifically, we can have an outcome of a drug base, a medication, in such that you can specifically block its action. This is the step that is critically important for the drug design, and that is the computational step. That is to take the three-dimensional structure of known atomic placement and trying to identify a spot where you could block things. And if you block its action of a protein that's associated with some kind of a bacterial infection or a tumor or some kind of ailment, then it leads to an applicable drug. Just to give you an idea of how much things cost during this, in the computer modeling stage, it's the cheapest. And this was done more than 10 years ago, and the prices have gone up. But this is just to show you that the lower end in computer modeling, in which we look at millions of compounds that may potentially block our protein targets that's associated with disease, are the cheapest. But as you go down this development trail, you have biochemical assays and looking at whether things that are determined in silico is accurate as well as in reality on the bench or even in the physiological system. So if we go down biochemical assays, cell cultures, and looking at how it affects the animal system, and finally in humans, which is FDA approved, then you could see the prices go up enormously. So this is why it's so important to get an accurate structure for structure-based design, because we like to get it at the lower cost end of this. And specificity is really important, because when you see medication or drug commercials out on TV, you see wonderful displays of all these outcomes of people taking these drugs, but at the end you see these horrible side effects that you see, and this is why specificity is so important. The structure base, in terms of how many structures are out there, is about over 100,000 to date. All right? And as of this morning, there's exactly 110,786 structures. What that means is that there are three-dimensional structures that are known. There's that many. But what I want to talk, what our application is what is called neutron crystallography. And this may be foreign to many of you, but neutron crystallography shows placements of hydrogens. And I won't go into details in terms of the chemistry, but if we look at the overall bank and how many structures have hydrogen knowledge or the locations of it, there's very, very few. There's only less than 100 structures for this. And just to give you a little bit about chemistry, in all proteins, and that's what we're talking about in most of the drug targets, hydrogen atoms makes up 50% of the atoms. So in which case, a lot of our structures that's out there is insufficient in terms of looking at drug discovery and using structure-based design. So what we are trying to do is use the ISS as a platform to grow large volume crystals. And to connect, what I'm talking about is large volume crystals is the bottleneck for neutron crystallography. Technically, you need large volume crystals. And for a lot of molecular reasons, it's very, very difficult to grow large volume crystals in the ground because of convection, mixings, and sedimentations, and so forth. And so we had flew an experiment that actually gave us some very important results that we feel that the ISS serves as a valuable platform for this endeavor. And modest, it's called the Granada Crystallization Box, or otherwise known as the GCB. The main containment, the main containment is here. Well, oops. Well, I'll just point to it. On your left-hand side is a cartridge of capillaries, and we fill up the proteins and we precipitants. These are reagents that hits the interface of the protein that induces in a metal canister, and okay, thank you very much. Containments which houses this whole experiment. So the whole layout looks like this. This has each cartridges that I've just shown you is displayed out here in these little bags. We flew about 20 of these. Each one of these about a diameter, 2 millimeter diameter volume of these capillaries. And again, I stress, we're out to grow large volume crystals. This is specific X-rays. And so this is the setup that we have these, all these packages that are laid out in these canisters, as GCBs. And again, they're under 3 levels of containments, and they're in a styrofoam. There's no temperature control or anything like that. It's very, very simple. This was delivered up to the ISS for about 6 months of time. So here are some of the results that we've got after 6 months. This was taken in the hotel room, and what you can see, this is with no magnification whatsoever, that large volume crystals are grown out to the diameter of the capillaries. We do not see this of those on the ground. And here's the reason and comparisons that we see. On your right-hand side are the conditions that were grown on the ISS, and on your left-hand sides are the equivalent that's grown on the ground. As you can see, the large volume crystals are prominent from those that are grown from the ISS, and not only are they prominent, they're very well separated in terms of the spacings, in terms of diameter of the data. If you compare these two, there's a drastic differences, at least from this particular enzyme that's associated with an infection. Here again is another shot. We have about 70 of these, and I'm just showing you a few as an examples of some of these remarked differences. Each of these is repeated 5 times, so they're statistically significant. So there is indeed a true effect due to gravity on these protein crystal growth. Again, on your right-hand side, same difference when, and we even tried to vibrate it with an equivalent, what we predicted to be the same impact as takeoff and re-entry. Here are some close-ups of the crystals, and for us, exceeding 1 cubic millimeters. These measure about 6 cubic millimeters. I know it's not big compared to a diamond ring or a small molecules that you're used to, but for us, this is a that used. So in terms of looking at quality of these crystals, these are typical graphs that show the left-hand side is the intensity. That is that how well they diffract off the neutron. So the higher the number, the better. And is you how resolved that you can differentiate the atoms. So the higher the number that you go out, the better, which is in overall resolution. So that means that you are able to definitively locate these atoms much better. The higher the resolution is like reading the 2010 on your eye chart on your doctor's office, and the low resolution means that you could barely make out the E in molecular standards. Here's another, and so here's the same thing that shows the crystal qualities of this. This is the electron density map to show the one we could trace the tracing of the atoms compared to those on the ground. And for neutrons, we're able to locate positions of hydrogens. Large volume crystals is essential for this. And this is my last slide showing in terms of what we can see in terms of space crystals that we're able to see positions of hydrogen. And in active sites, these are critically important if we are able to design drugs, ligands, targets to block specific sites for a particular protein. In terms of providing opportunities, and we look forward to extending our experiments more on this. So thank you very much.
Very much. Next, Dr. Look.
We're developing new crystals and sensors for radiation detection. The applications vary from medical imaging, homeland security, high energy physics, low energy rare event physics, and we collaborate with IIT, NASA Marshall, and TechMasters in this cases funded program to grow crystals of click, cesium, lithium, yttrium chloride. It's a new scintillator which is sensitive to both gamma rays and neutrons. So that's what makes it unique. One sensor can be used for detecting both neutrons and gamma rays, and it's also spectroscopic, which means it can detect not only presence and intensity of radiation, but also identify the energy, which in turn leads to identifying which isotope is emitting the radiation. So we supply this, we developed this click crystal a few years ago, and it's a product for sale at our company, and we supplied for several different instruments made by Thermo, and also for a variety of different instrumentation builders. So highlights of click features: it has bright response and high efficiency for neutrons. It allows for pulse shape discrimination for gamma rays and neutrons using different rise and decay times for gammas and neutrons. And it has a good proportionality, which results in a good gamma ray energy resolution less than 4% at 662 keV. And it also allows for fast neutron detection due to presence of chlorine-35 isotope. This is how the pulse shape discrimination works with click. You can see that gamma rays and neutrons have different shape of the pulse, and using processing analog and digital processing algorithms, it's possible to distinguish between those and differentiate between neutron and gamma ray events. However, even though a click is already a commercial product, we still have a number of challenges in crystal growth. In particular, the defects in click crystals can be grain boundaries, inclusions of secondary undesired phases, and cracks of the crystals. The goal of this project is to grow several click crystals in microgravity. The biggest advantage of microgravity for crystal growth is absence of buoyancy-driven convection, which totally changes how the mass transport works in the melt and how the crystals work. So the segregation of components of click, which is a mixture of three components, cesium chloride, lithium chloride, and yttrium chloride, is initially more uniform because there is no density-driven segregation. Because there is no convection, the segregation at the solid-liquid interface is controlled by, also weightlessness of the melt results in eliminated melt trapping between the crystal and crucible and reduced cracking. This is the hardware furnace that we are going to use on board of the ISS. There's an identical unit on the ground that we are using for ground-based testing currently. It was designed and built in 2002. When I was a graduate student at RPI, my professor, Alex Ostrogorsky, had a NASA-funded program to grow crystals of indium antimonide using this furnace. NASA, in cases, wanted to continue using the equipment, and we are using basically the same geometry of ampules and same furnace, which was recently upgraded by TechMasters to more modern controls, but the hardware stayed the same. In the frame, we were able to monitor crystal growth of indium antimonide alive as it was growing, and because the furnace had a transparent section here on the right. And this furnace goes inside a microgravity sciences glove box over here, and there's a camera looking at it. The purpose of the glove box is to protect astronauts in case breakage of the ampule and crystal material spilling out. In the picture here, Peggy Whitson in 2002 is working, astronaut Peggy Whitson is working on one of our experiments. So we developed two configurations for ampules that would grow click crystals in space, and we will also conduct a series of reference experiments on the ground in identical geometry. So basically there are two configurations with seeded growth and unseeded, where we'll be using a piece of click single crystal as a seed or a nucleation capillary, which is like we do grow crystals for production. These are the flight ampules that we recently prepared. The gold coating, the purpose of the gold coating is to reduce heat losses from the crystal and the ampule, because the melting point of this crystal is higher than indium antimonide, for which the furnace was originally designed for. So in summary, RMD developed this very promising and commercialized, very promising click scintillator crystal, a first scintillator used for both neutron and gamma ray detection. So far, there have been no safety issues during ground-based testing, such as ampule cracking. Also, crystals don't crack under higher temperature gradient compared to our production process. And our goals are to understand mechanisms of defects formation in click crystals without interference of gravity and buoyancy-driven convection. And our ultimate goal is to improve production on Earth. Thank you.
All right, next, our final speaker in the panel is Dr. Roz Smith, and she's from Lilly. Yep, exactly. She's coming up imminently. We're swapping out computers here in order to be able to facilitate this. And while we're waiting just to swap this over, we will have again 3 questions for our panel right after Roz's briefing, and then we'll open it up to these will kind of be lightning round, hard-hitting questions, and then we'll open it up to you all. Thank you.
Good morning, everybody. In this brief little time that I have, I'm going to try and tell you a little bit about how we've been using this year the ISS platform to aid some drug discovery efforts for muscle wasting diseases on Earth. And it's not just astronauts that undergo muscle wasting, obviously, in space, but we also know that that muscle wasting actually is an issue, a medical issue here on Earth. For actually, there's over 100 different conditions and diseases that have been associated with skeletal muscle wasting and atrophy. And it's not really just the decrease in muscle size that matters, it's actually the fact that when you have smaller muscles, you have reduced muscle function and are weaker. And it's that sort of weakness, muscle weakness, that's really critical to the patient, because when you're weaker, you can, in certain situations, not able to perform activities of daily living, climb stairs, for example. You may be, because you're weaker, more at risk for falling, fracturing. It can lead to, particularly in the elderly, nursing home admittance. It can lead to lack of independence. And so you can understand that muscle wasting and muscle atrophy and decreased muscle function leads to diminished functional performance and is associated with poor prognosis and outcomes. And unfortunately, the only real treatment for such conditions is either physical therapy or exercise, which, if you're ill or elderly, is often there's poor compliance and often not enough, not sufficient. So drug companies have actually been thinking about this now to try and think about developing a drug to combat muscle atrophy. And some drugs are working their way through clinical trials, but at the moment still nothing's got FDA approval as yet. So mouse models of muscle atrophy. The mouse actually is a great model organism for studying muscle atrophy. And when we're developing drugs, we always have to have some preclinical data to support clinical development of any new compound. And the mouse is, as I said, a good model organism because there's a lot of conservation of just the biology, the cell signaling, and the key genes involved in muscle wasting compared to man. But as we know, mouse models, terrestrial mouse models of muscle atrophy are not ideal. We've already heard a little bit, I think, in the meeting about hind limb suspension model. But if you're aware of that model, that unloads the hind limbs, but the forelimbs actually have more weight on them. So it's not an ideal model. And in microgravity, you have the ability to unload all the muscles and therefore have more of a global model of muscle wasting, which actually is more akin to a lot of the actual muscle wasting diseases that people have here on Earth. So we were excited this year to have the opportunity to explore the microgravity environment to under rodent research 3 to determine if one of our novel compounds was able to prevent microgravity-induced muscle wasting. And of course, our goal was to use the results to help find ways to develop new medicines for patients in need with muscle wasting conditions. And so the experimental design was, and we flew this experiment on SpaceX 8, it was launched in April, was that we had 10 baseline animals, then we had 20 mice go up into the ISS, 10 were controlled treated, 10 were drug treated. And of course, we had a similar number on the ground that were our ground controls, where everything was sort of identical except for the microgravity factor. They were female mice, 12 weeks of age. We did a 6-week experiment. And as I said, fortunately, things went well. They launched. And one of the things that we wanted to do for the first time in this experiment was not just look at muscle mass, but look at muscle function on board the ISS. And it's possible to do that in mice with a grip strength meter. You actually just measure the ability of the mice to grip on. And we set that up and used that. We use it on Earth quite well, and you can get significant effects with this grip strength meter. But we did that for the first time on the ISS. And I'd like to share the results of that those data with you. So this is the time point of, and we did an interim time point at 4, and 6 weeks was the termination of our experiment. And this is the % change from baseline in grip strength. These are our control animals, untreated animals on Earth. These are our animals in the ISS. You can see they reduced their ability, their muscles got weaker. And these are the ground controls treated with our compound, and you can see they got stronger. And in green are the flight animals treated with our compound, and you can see there was a significant increase in their strength, actually getting back close to the untreated ground controls. So that was very interesting that we were able to prevent some of that microgravity-induced muscle weakness. We were very fortunate that our samples at the end of this experiment came back to Earth in late August on SpaceX 9, and so we were actually able then to actually measure the muscle mass of these animals. So I'll be sharing just a snippet of those data, which we're still working on, and that's the muscle mass data from this experiment. And I picked 2 muscles just to show you today in the interest of time. The gastroc, which is a muscle in the hind limb, and the triceps, which is a muscle in the forelimbs of these mice. If you look, you can see that this is again changed from baseline. The mice are growing, so there is a change in the ground controls. They actually increased their muscle mass during that 6-week experiment. But you can see the flight controls, there's a decrease in the muscle, of this particular muscle, during the microgravity experiment. And here's the effect of our compound on this particular muscle. And you can see that we got the expected increase in our ground controls, but in the flight, we also got the increase, and it went up to the level of the untreated ground controls. A similar pattern here in the triceps, a decrease, not statistically significant, but a trend for a decrease in the flight animals in this particular muscle, and our compound was able to still work in the conditions of microgravity in a very similar manner to that in the ground controls. So obviously we're still analyzing the data, but we have some evidence that our compound was able to prevent the microgravity-induced muscle wasting, including the decreases what we saw in muscle function in microgravity. We're still analyzing the samples and data. We're still getting data and still working on that. And of course, the data will be used to understand the effects of microgravity on muscle, but also to help our drug discovery efforts to find drugs to combat muscle wasting diseases. And we also do plan to, we shared a lot of the samples from the mice with other investigators, and we plan to share, to publish the results. Takes a village to do this experiment. We have lots of people to thank, haven't got the time, but all our collaborators are mentioned here. Thank you.
Okay. Let's see if you can hear me here. All right. Well, we're going to go very quickly to, and since since we have about 4 minutes left in the session time, we'll actually go a little bit longer since there's no one immediately following. However, the focus of the panel, remember, was research enabling LEO commercialization. So you heard about crystals, integrated circuits, monitoring detectors, bioregenerative solutions, all that are helping to transfer or truly benefit life here on Earth. So here's a couple of questions within that context. Number one, opening this up to the panel, do you believe that we are an inflection point in commercial space R&D, and if so, why?
I'd like to answer maybe not specifically whether, because nobody knows for sure whether we are at an inflection point, but an inflection point can come anytime when there's a shortage of funding by government. And so currently most of the ISS projects are funded by the government, and we want to make it more self-sufficient by commercializing and having companies pay for it ultimately. So I just had an idea of how about a program where companies would have to pay some kind of a copay, which would be subsidized, which would be an incentive to for commercial companies to use it, and the amount of the fraction that the commercial companies would pay for the research would be dependent on the demand. If there's a lot of companies who want to use the ISS or another space LEO vehicle, then the copay would go up. If not enough, then it would go down. And another important factor, I believe, is having more universal tools available on the ISS. For example, in our project, we are using furnace and MSG glove box that were designed over 10 years ago, and we still can use them. So having more tools or equipment on the ISS, which can be used for processing new samples, new materials, using the available facilities would also be helpful.
Okay, thank you. Anybody else want to weigh in on that one?
I'll just add on really quickly. I don't think we're quite at the inflection point, but I think we're close. And the reason is that I think a lot of the scientists in both commercial and academic sectors are still having a risk-averse mindset. And that could be from economical reasons, budget funding and the universities in terms of, making sure that things are working. But I say we're close to the inflection point because I agree with my colleague here that there has to be some incentive to provide us to get over that risk, that risk-averse mentality. Because I think we're getting to a point where we're intrigued to see results coming back from space. For example, in muscles, in chips, crystals, and things like that. And this is by evidence of the grant agencies like NIH and NSF getting involved with supporting studies associated with the ISS. And so that is why I think we're close to inflection point, but I think we need more federal funding, perhaps, but more commercial to jump on board and say, we're willing to take that risk because we see results coming out.
Sure, thank you. So the follow-up then is and you've touched on two potential solutions. What do you see as the single greatest challenge or hurdle for sustainable LEO research from the commercial sector?
Well, I can make a few comments that I still think. We're very excited by the results that I presented in terms of the ISS and what we can do in that for example, muscle wasting diseases. But it's still obviously it was a subsidized experiment in terms of cost. Costs are still, if we had to have paid the whole amount, would I think been prohibitive. So we still have to think about how to make that more affordable, and also the time scale in drug discovery. We have to work on a shorter time scale than waiting a year or more to think about the experiment and get the results. And of course, our experiment was affected by delays because of rocket explosions, etc. So somehow we've got to get that into a more affordable, shorter cycle times, I think, to make it really, work and effective.
Excellent. And kind of a derivative from that is really, as we look at the value proposition for space-based or LEO research, is it, each of you showed how your experience in space was truly revealing or beneficial, but relative to other competing R&D alternatives that are terrestrially based, do we yet have the value proposition for space-based sustainability, in your opinion? Supriya, you got any thoughts on that?
For me, sustainability is about sustaining a community. Can you support, human life in space? Can you support a community in space? And to really get to that aspect, my belief is that to really have a community that you can sustain, you have to be able to take advantage of raw materials that are already out there and be able to use those effectively. And when we get to that stage where we understand how to use raw materials, and also we understand how to commercialize using those raw materials in space, that's when we'll get to this stage of sustainability. And I think also hand in hand with sustainability comes safety, because many of these raw materials sort of are based in oxygen-deprived kind of atmospheres, and it's understanding how we can use those raw materials given the constraints of not having oxygen out there. So once we come to the understanding of those two elements, that's when we can support sustainability. Great.
Okay, we have some more questions here, but I'd like to open it up to the audience to see if we have anyone. Yes, sir. Sure. Oh, okay, sorry.
Yes, thank you for this interesting session. I'd like to clear up one comment on the hind limb suspension model. I'm Alan Harkins from University of California, San Diego. I think Dr. Lewis indicated that although there's lower than normal weight bearing on the hind limbs in the hind limb suspension model, she indicated there was more than normal weight bearing on the forelimbs. So we've actually measured forelimb weight bearing, and it's absolutely normal. So what's the other half of the body is taken up by tail tension. So I'd just like to make that comment that the hind limb suspension model is better than what was said at this session. Thank you.
Okay, any other questions? Yes.
I needed a secondary catcher here. So thank you for all coming and taking time to share. I think that the issue you highlighted about us being at the cusp of a very highly subsidized access is a really interesting time in the industry. And I'm curious, particularly for pharmacology and for the chips industries, these are massive multi-billion-dollar pipelines that you're entering into. How do you view the evolution of coming into space station at completely subsidized rates like your early access and moving towards a more commercially sustainable model? How do you think that works? Is it a tapering scale over time? Is it something that as you make your first billions that you pay back to the space station? How do you envision that evolution happening?
I think there's a very tough balance for the commercial sector on whether to report positive results as well as negative. And in terms of this is a catch-22. People would not get on board to utilize space station as a platform unless they see success. But we get success, but as a commercial sector, we possibly cannot reveal everything, for drug designs and so forth. So I think somewhere along the line, we're going to at least have to reveal more positive results without giving away the company, I should say. Because I think the important thing here is people is got to see some more success. This has been the problem in the last couple of decades, that microgravity is you don't get the bang for the buck because you don't see as much positive results as possible. But there are a lot of positive results, especially now, and you've heard them here. But in terms of publications, things like that, it's still very limiting. So I think as a community, especially speaking for the private sector, I think it would do the community for usage ISS much better if we start revealing, or at least publishing, things that will showcase the positive outcome of the ISS without revealing a lot of the commercial secrets.
But Joe, would you be willing to pay $7 million for your payload?
I'm sorry?
Would you be willing to pay the full $7 million that it takes to take a payload to space?
If I had $7 million, I would be willing to do it.
Right. So I guess the question really is down that particular axis. Right now, access is highly subsidized. Obviously, for us to be commercially sustainable, it needs to be monetized. So how do you envision that changing?
Well, I guess what I'm trying to say is that if we're being subsidized, I think we should have a responsibility for justifying the subsidy. And like I said, there are many ways to do it without selling out the company. In my field, in terms of crystal growth, I think I could talk all about how the difference with getting large crystals without showing atomic coordinates for structures of drug design, for example. And right now, we're not doing that. And I think that we're seeing so much results that more people will come on board and say, wow, there are lots of positive results coming out of that.
How about anybody else? Because I think this is the $100,000 question. I mean, that is truly, are we getting to a point where we don't need the large-scale subsidization by the U.S. government or by governments in order to enable a vibrant space-based, LEO-based R&D ecosystem? Or, when do we get to the tipping point where it's self-sustaining? Is there a mix among those? And I'd love to hear any more thoughts on that, because I think your point is right, but I think it's on the periphery of this $100,000 question, maybe $1 billion question, $100 billion question.
I'd like to add to that point as well, if that's possible. There's an economics argument to be made, and it's really this: are you operating in an industry where it's more beneficial and more cost economical to put it in space than it is to do it on Earth? And the electronics industry and semiconductor industry is a $400 billion industry and a sizable proportion of GDP. And it turns out that when you make one product, and one product costs $150 million, it is definitely cheaper to pay $7 million and put it in space and access the raw materials there than it is to try to build a $150 million tool on Earth. And it comes down to two things. Is it really the cost of just bringing it to space, or is it the cost of operating in space? And if the economic argument is there that it is cheaper to operate in space than it is on Earth, you can sort of justify the cost to bring it to space. But you have to balance both of those two aspects, and you should pick big problems to solve. So that's really the other argument. Is it so important that we have to do this in space that it will really advance and accelerate and benefit life on Earth and make the economics argument cheaper on Earth?
Now, I have a question to Supriya. Then, who makes the judgment how beneficial the research is, if it's a commercial product or commercial project?
It's the same judgment that goes into who reviews the proposals that justify whether a project should be launched on the International Space Station or wherever. It is, there's a peer-reviewed body, a collection of scientific thought that really believe that this is the future and this is how we should accelerate to get to the future.
And piggybacking on that, you know, you're the CEO for Astrolux. You know, you're the head of the company, so you're making company bets on that as part of your fiduciary responsibility and part of your overall activity. So I think that's a fundamental driver. I mean, you're making a value-based proposition or a decision based upon cost-benefit in the fully reviewed portfolio. So I think that's an important element. How about any other questions? Because this is definitely good. Yes.
Yeah, this is a question for Dr. Smith. I understand that Lilly's drug was originally developed using animal models of some sort to establish the original efficacy of the drug, and now you have a space-based test, and I was wondering how your original animal model results compared with the space-based test, and if the space-based test were available in the original development of the drug, if that would have speeded up your pipeline or had helped your decision making.
We're still processing the data, but so far, as you can see, the results in space actually are somewhat similar to the ones we see on the ground, except that the models of wasting, as I mentioned, on Earth, have some limitations. So it would have been lovely to have, in the past and for molecules in the future, to use that environment as an ideal way of looking at effects of compounds on muscle wasting. But as we talk about in terms of value proposition and cost benefit, I think one has to still be thoughtful in terms of, as I say, the time, the cost, when actually it's still one piece of a puzzle, to put a whole package together to be able to get a drug to even go into the clinic is multifaceted, and the one piece of preclinical data is only one piece of that puzzle. So yes, it would be nice, but as I mentioned earlier, I think still the time, the cost would not be is not sufficient enough value at the moment on its own for us, for example, to pay the full cost of it, I think was one of the questions. But certainly we're starting to appreciate the value and have the opportunity to look at that and appreciate the value of these experiments and that environment can bring to preclinical drug discovery.
Any other audio? Oh, great. Yes, Sam.
Hey, John. I'm Sam Shamimi, the director for the space station up at NASA headquarters. I've got a couple thoughts on this subsidization of access and use. I think from what I've seen, access is really a big issue, and we know that on our side. And the limitations on board the space stations, crew time and the like, are also real limitations. But I think when the supply side, when the demand side, when research companies or production companies or companies that are heavily invested in mass production of whatever, when their demand really outstrips what the government can do on its own, that's when I think the subsidies will have to be addressed. Because again, if your access is now limited by government provided capabilities, but your market is demanding that you do more research in space, then I think that's when industry will want to start investing, in, either to augment what the government is doing or to have their own access and capabilities. Any one of you, would you like to like project in the future? I mean, what would it take for your industry to be at a point that it would be willing to at least start to make investments on the supply side of this business?
I agree with your statements about how it would transition, and I think an important factor to motivate commercial companies is marketing, because a lot of companies in the industry, unless they are closely involved in space and microgravity and ISS research, they don't know that such capability exists for them to do their work on the ISS or any other low Earth orbit vehicles. Traditionally, the model has been that NASA, or more recently cases, they can come out with solicitations and researchers come to them proposing to do some research work justifying how important it is. In this case, it could be beneficial if government agencies in cases would outreach to industrial community, advertising those as a fee-for-service facility.
Okay, I think that will lead us to our final question, which is, as we go into this big presidential transition, as we look at policy drivers for this and shape that future, what one policy or action would stimulate the most investment and activity in space by your company? Is there anything that if you had a wish, you would wish for X in that policy or action? And we have many in the government audiences here, as well as many in the industry side. Any thoughts? Okay, we'll take that to ponder and think about. I think what you've heard today is 4 really shining, vibrant examples. You've heard crystals, ICs, monitoring, detecting, biopharma, bioregen, very hot areas, very capital-intensive areas, but also huge market-based drivers. So I hope that this was a thought-provoking set of discourse. You got to see snapshots of real results, but also talk about the potential that each of these is a microcosm of the broader LEO space economy. So with that, let's please give our panelists a round of applause and thank you. Okay.